Hello everyone, welcome to today's live broadcast, Proteome Profiling of the Tumor Microenvironment, Role of Human Primary Fibroblast-Derived Exosomes in Oral Cancer Progression by Dr. Simona Principe. I am Dr. Chad Schwartz, Senior Application Scientist at Beckman Coulter, and I'll be your moderator for today's event. We are delighted to bring you this educational webinar presented by LabRoots and sponsored by Beckman Coulter. Beckman Coulter is a leader in centrifugation and flow cytometry and has long been an innovator in particle characterization, lab automation, and genomics. Beckman Coulter products are used at the forefront of important areas of investigation and discovery. For more information, please visit beckmancoulter.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box at the lower left of the presentation. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the icon in the lower right-hand corner of the slide. If you have any technical problems viewing this presentation, please click on the support button at the top right of your presentation window or submit your problems to the green Q&A button. I would now like to introduce today's speaker, Dr. Simona Principe. Simona completed her PhD in immunopharmacology in Italy, and she is currently conducting postdoctoral research in the lab of Dr. Thomas Kisslinger at the Princess Margaret Cancer Center in Toronto, Canada. Her research interest focuses on applying proteomics technologies to investigate cancer biology. In particular, she studies the role of secreted extracellular vesicles as novel intercellular signaling mediators within the tumor microenvironment that support cancer progression and metastasis. Since 2012, she has published 5 papers and reviews in the field of proteomics and exosomes. Before we get started, I would like to ask a few poll questions. Our first question is, what is your current isolation method for extracellular vesicles? Please select multiple answers. Differential centrifugation, density gradient centrifugation, immunoaffinity capture, other, or I don't isolate EVs. We'll take a few moments for the answers to populate. It looks like actually the majority of folks in attendance do not isolate EVs. The 26.5% use differential centrifugation, 5.9% use immunoaffinity capture, 2.9% at density gradient centrifugation, and 2.9% at other. Our second and final question. How do you analyze the purity of your EVs? Again, please select multiple answers. Just a few moments to allow the answers to populate. All right, it looks like 35% actually do not check for purity, whereas 19% use nanoparticle tracking analysis, 19% use electron microscopy, and 13.5% use protein absorbance ratio, and another 13.5% use dynamic light scattering. Thank you everyone for your answers. I will now turn it over to Dr. Principe for her presentation.
Thanks, Chad, for the very nice introduction, and thank you everybody for taking the time today to listen to my webinar. So today I'll be talking about some proteomics analysis that I've been conducting in my lab of the tumor microenvironment, and with particular focus to the role of human primary fibroblast-derived exosomes in oral cancer progression. This work is currently ongoing at Princess Margaret Cancer Center here, University of Toronto, and this is my postdoctoral fellow project under the supervision of Dr. Thomas Kisslinger. So just to give you a little bit of background on head and neck. Head and neck cancers are is a group, a very heterogeneous group of cancers that includes different tumors, the races above the collarbone, and the 3 major subdivisions are oral cavity, larynx, and pharynx, within the oral cavity being the most prevalent. As you can see here from the list, each one of these sites also had multiple subsites. So in this case, particularly for this talk, I'll be focusing on the oral tongue, which is the source of my samples. The major risk factors for head and neck are alcohol and tobacco consumption, and together account for 70–75% of cases. Despite few progresses in surgery and chemoradiotherapy over the last 10 years, I would say, still the 5 years overall survival of these patients hasn't really improved. So this actually suggests that there is a strong need to better define the molecular basics of this cancer. And also the incidence rate are higher in men as in women. From a pathological point of view, most of this cancer, 90–95% of them, are squamous cell carcinomas, also called SCCs. And the remaining 5% instead accounts for less common histologies. They're usually variants of SCCs. So we can classify the tumors based on the percentage of keratinization of the tissue. And we can see here an example of normal epithelium, as well as the well-differentiated, moderately or poorly differentiated tumors. That's the latest stage. For head and neck cancer tumor development from an histological point of view, we can see how the normal appearing epithelium turns into an hyperplastic and then evolves to a mild dysplasia. Then turns into severe dysplasia, also called carcinoma in situ. And then the latest stage of this cancer is the invasive carcinoma. That's so-called invasive because it becomes usually metastatic. So in parallel, at the molecular level, what happened in the condition of normal epithelium, you can see that the epithelial cells are very well separated from the surrounding stroma, but by an intact basement membrane. So the underneath stroma contains different cell types, cellular and non-cellular components. Among the cellular components, there are different components of extracellular matrix, as indicated here by the collagen bundles, for example. And among the cell types most common in the tumor stroma, we have immune cells, leukocytes, and as well as endothelial cells and fibroblasts. So I will focus my attention on fibroblasts today, because what happened when the normal epithelium start turning into a condition of mild dysplasia, you can see how the fibroblasts that initially are quiescent in the stroma start becoming activated. And this activation changes and modifies their phenotype. So still you can see that the epithelial cells, here indicated in blue, are proliferating. That's why the condition is called dysplasia, but they're still very well separated from the stroma by an intact basement membrane. So what happened next when the tumor evolves to invasive carcinoma is that the activated cell types, such as the fibroblast, in this case, of the tumor stroma, turns into myofibroblast phenotype, and it starts secreting numerous amounts of molecules and extracellular matrix component that contribute to the stiffness of the tumor, as well as enzymes that degrade the basement membrane, and they lead to a condition of mixed cell type together. So this is the condition of a stromal activation, where we have, as I mentioned, degradation of extracellular matrix component, infiltrate of immune cells as well, and the differentiation of fibroblasts into CAFs. CAF stands for cancer-associated fibroblasts. So the origin of the cells is still controversial. One of the most popular hypotheses is that, as I just mentioned, they are generating by paracrine activity of cancer cells. So epithelial cancer cells that, for example, secrete TGF-beta or other interleukins that actually act as a paracrine signaling on the fibroblasts and activate them. Or other hypotheses also support the transition from totally different cell type, as for example, epithelial cells, they undergo EMT, or pericytes or endothelial cells or other cell type more. Then they're also probably deriving from mesenchymal stem cells. So cells that come from the circulation that in this case have entered the tumor site due to the leaking vessels of the late stage of the carcinoma. And from a phenotypical point of view, how do we differentiate a normal fibroblast from an activated fibroblast? So the CAF phenotype is mainly characterized by expression of smooth muscle actin, actually different isoform, but the gold standard marker is the alpha-SMA. And this is actually contributes to the contractile phenotype of the cells that don't look affusilated anymore, but they look more spread. And they also have a greater rate of ECM, synthesis and secretion, and they're resistant to apoptosis. So numerous studies recently have taken into account the importance of the tumor microenvironment in tumor development, because the stroma actively modulates the biology of the tumor by acting on the cancer cells. The interaction and the paracrine signals between stromal cells and cancer cells are bidirectional and extremely dynamic. And CAFs are a key component in regulating carcinogenesis. So starting from the hypothesis that the stroma can promote tumor growth, my main project has been focusing on isolating CAF, cancer-associated fibroblasts, as well as adjacent fibroblasts that from now on I will call AF, from resected tumors that derive from tongue. And here you can see those are OSCCs, that stands for oral squamous cell carcinomas. So after isolating those cells, I have done an in-depth proteomics analysis of two different pathways, canonical and non-canonical secretion of proteins. So I've looked at soluble proteins that are secreted by the fibroblast and released in the media, as well as I've looked at non-canonical secretion of exosomes. What are exosomes, and just briefly, what's the role in cancer? Exosomes are nanometer-sized extracellular vesicles that range from 40 to 100 nanometer usually, and their origin comes from the multivesicular body formation. This is where their biogenesis come from. We can see that where stimulus acts on the cells, there is the formation of intraluminal vesicles that then turn into early endosomes. The early endosomes mature into late endosomes, also called MVBs. This stands for multivesicular bodies, because it's bigger vesicles that to have content that contain a smaller nanovesicle inside, and then the MVVs can undergo lysosomal degradation or can fuse with a plasma membrane and release their content in the extracellular space. So in this case, the content released will be the exosomes, and so the exosomes are released from different cell type, and thus they can be found in body fluids. They're also being used as biomarkers, and they're involved in the intercellular communication among different cell types. What's inside the exosomes? So they contain a variety of different molecules ranging from protein component lipids and also nucleic acid. Exosomes has been quite well characterized lately, and they also have membrane markers such as tetrascanning and CD molecules that are used as common markers to define different vesicle populations. And so lately, they have been described in numerous amounts of papers. Exosomes' role in cancer has been shown to have effect on intratumor heterogeneity, for example, on immune system escape and inhibition, and also having effect on angiogenesis and metastasis, as well as in the transformation of the fibroblast into cancer-associated fibroblast. Regarding instead the exosomes, they're secreted from the fibroblast and acts on the tumor cells, so the opposite signaling that's object of my study. There's two main papers that recently came out, and they're worth a mention in this field, and have been also co-author of the Nature Cell Biology one. Basically, both papers shows in different systems how CAF-secreted exosomes have an effect on cancer cells, and different type of cancer. For the South paper is breast cancer system, for example, and this activates signals that induce cancer migration and also angiogenesis. So particularly for this talk, the main aim has been to uncover fibroblast proactive regulators of tumor progression. And I've shown you that this has been done by liquid-soluble proteins as well as exosomal proteins. So this main aim has been done through the 4 main different steps. So the first aim was to isolate and characterize primary CAF and AF that has been isolated from resected tongue cancers. The second aim has been to conduct in-depth proteomics analysis of CAF and AF secretome and exosomes, then evaluated the effects of fibroblast-derived exosomes on tongue cancer cells. And lastly, I've also been generating a signaling map of fibroblast cancer cells ligand receptor interaction in order to complete the picture and sort of close the loop of this proactive signaling. So going through aim 1, the isolation of human primary CAF and AF has been done from resected tumors that come from the tongue, so primary patients. Those have been dissociated into single cells by using a mixture of 2 different enzymes, collagenase and hyaluronidase. Then this cell suspension has been further resuspended in a red blood cell lysis buffer, and the cells have been seated down on plate. So for fibroblasts specifically, the only cells that are going to adhere to the plate, in this case is the plastic, to just a plastic petri dish, are the fibroblasts, and then the other cell type gets washed off. And of course, on the cells, then it gets evaluated for positivity to fibroblast markers and negativity to, for example, CD31, endothelial cell marker, or the epithelial marker, etc. So what happened in parallel to as light of the tumor get also undergoes histopathological evaluation. And so in the end, I get also some clinical info on each patient, and I have a final table that gives me also information about the site, differentiation stage, and tumor stage. So the first aim after having isolated the primary human fibroblasts from the tumors has been to characterize and make sure that the in vitro system that I'm looking at recapitulates actually what happened in vivo. And I started by validating this for the gold standard marker, which is half SMA. As indicated here in the Western blot panel, both panels show the expression of alpha SMA, which is higher in CAF versus the adjacent fibroblasts across 9 pairs that I've been able to isolate from tongue tumor. And here in the second panel, we're looking at an immunofluorescence still from alpha SMA, and we can see how the morphology of the CAF, it's absolutely different from the adjacent fibroblast, and how the alpha SMA actually defines this contractile fibers there within the cells. So the first 4 pairs have been the pair that I will call from now on discovery cohort, because those are the samples that have been further validated and then used for the proteomics analysis. And you can see here how I also looked at their morphology, and the morphology looks different. The AF are more elongated, whereas the CAF are more spread. Especially this can be appreciated when they're seated at low density. And also I've been conducting a functional assay, which is a collagen 1 gel contraction assay, where basically the cells, the fibroblasts, are embedded in a semi-solid matrix of collagen 1, And then I measure over time the activity and the capacity of the CAF to degrade and shrink the semi-solid matrix of collagen. So here in the graph, you can see how at 6 hours, basically the collagen area, it's reduced for the CAF marked in red versus the adjacent fibroblasts, which is the blue dots. And then the second cohort of patients, the remaining 5 have been used as a validation cohort for more of a functional assay and validating the biology after the proteomics funding. So specifically, going into aim number 2, the shotgun proteomics workflow, which is the strategy that we applied here in the lab and that I've been optimizing for this cell type, consists of protein extraction from CAF and AF. And for this, we use TFE, which is an organic solvent, actually extracts the really well-membrared proteins. Then proteins are digested into peptides by using trypsin. Peptides are separated by ultra-high pressure liquid chromatography. And then they're analyzed into a mass spectrometer. And here in the lab, we'll be using a QE. So the final data is analyzed by using a MaxQuant platform and searched by Andromeda algorithm. So, and this will allow us to have a semi-quantitative idea of the proteins in our list by using peak integration method connected to the software. So after that, of course, more bioinformatic filters and more data mining is being applied to the final list of proteins in order to deal with more manageable list of proteins and also to find some enriched protein signatures that we're interested in looking at. So a little bit more in details into the protocol that has been used for proteomics. I've been working on 4 different pairs of CAF and AF, so for a total of 8 cell lines. From each of them, I've been analyzing 3 different fractions: the whole cell lysate, the conditioned media, and the exosomal fraction. The whole cell lysate is being processed, as I just mentioned, by TFE extraction, whereas the media has been further concentrated and the exosome has been isolated by a standard differential ultracentrifugation protocol by using a Beckman centrifuge. And then all those 3 fractions, so that's a total of 24 different fractions, have been run in triplicate and analyzed by LCMS. And for a total of 72 runs, that gave me a final list of proteins identified and quantified of 4,247. So this is my final data set. And to just start a little bit more of data mining on these proteins, I've been looking at soluble molecules released in the media that are known to be enriched in the CAF versus the adjacent fibroblast. And as we can see here, in this box plot, different kinds of collagen 1 are highly secreted by the CAF versus the adjacent fibroblast is significantly enriched in the media of the cells, as well as the secretion of extracellular matrix component. And here I'm reporting elastin, fibronectin type 3, and SPARC. And also I've looked at the exosomal fraction to validate it further, and here you can see in the first image a representative pictures of electron microscopy analysis after our isolation protocol. And in the bar chart instead at the bottom, we can see how when we look at protein intensity of exosomal specific markers here shown in red, those are enriched, whereas cytoplasmic markers that are belonging to different compartments within the cells are depleted in the exosomal fraction, as we expect. So moving forward to more in depth into the data, after having this heat map shows, summarizes all the final data. And this is based on a unsupervised hierarchical clustering. So the way to read it is the highest is, the darker is the color, the most abundant is the protein. What we learned from this heat map is that the three fractions that I have been analyzing have split nicely between each other. And you can see also that there are different clusters that show up. For example, the light blue at the bottom, it's more of a background cluster that contains high-intensity proteins that are present in every fraction. But there are also, we can get also nice information if we want to look at a single fraction. And this is the case of the light green cluster more at the top. Instead, that we can see it's more enriched for proteins that are present in the host lysate, but not present in the conditioned media or in the exosomes. And then after this, I would just focusing mainly my attention on the secretome and looking at the conditioned media and the exosomal fraction, and more using the host lysate as a background all the time. So the first finding that I've noticed by looking at this data is that exosomes are actually enriched in proteins. They're involved in the regulation of cancer cell migration. And in fact, if we look at this heat map that represent a shorter list of proteins that are actually contained in the gene ontology analysis under the regulation of cancer cell migration term, we can see how 70% of proteins are enriched in the exosomal fraction versus the conditioned media. And the way to look at this heat map is exactly as the previous one. The darker color means the protein is more abundant. And if we zoom a little bit more into this proteins that are almost barely present in the conditioned media, that comes up to be a list of 151 proteins. And this list is reported here in this view, more of a zoomed in view. And this list is actually contains lots of oncogenes and proteins that are known to be involved in cancer cell proliferation and progression, such as HRAS, EGFR, or BCR, for example. And then the last thing I want to point out about this image is that visually, if we look at truly the distinction between CAF and AF, as indicated at the top of the map, we don't see visually a great difference in the protein expression. But I was able to go manually through the list and select a few candidates. There are actually a couple of candidates in this case, endoglin and ICAM1. There are enriched in the CAF exosome versus the adjacent fibroblast, and I've been validating this in a different cohort of patients in the exosomal fraction, as you can see here in the West blot images. So this first analysis suggested me that fibroblast exosome might contain key signaling proteins that are related to cancer progression and specifically cancer cell migration. So to test this, I first wanted to validate that the signaling, the paracrine signaling, and I've been treating the SCC25, which is an oral cancer, actually tongue cancer, commercially available cell lines that's immortalized and stable. I've been treating the SCC25 with exosomes and been just evaluating the uptake by marking the exosome with a lipophilic fluorescent dye. And we can see at 6 hours, we can already see a green signal of uptake, and this gets stronger at 24 hours. So after having verified this, I wanted to mainly evaluate the effect of exosomes on cancer cell migration. And so for this, I used pretty much the same setup. I've been treating SCC25 with the exosomal fraction as well as with the EFM that we can see here as indicated in red. The EFM stands for the exosome 3 media, which is basically the supernatant that come from the after having spanned down the exosomal pellet. So it doesn't contain vesicles. And here you can see in this panel a plot for flutilin-1, which is an exosomal marker, and that's just to check the purity of the two fractions. So flutilin only shows in the exosomal fraction across 2 different patients analyzed and reported here. And then what happened when we treat SCC25 with exosomes, We can see here a representative image of 7 different patients that have been analyzing so far, where the exosome treatment enhances cancer cell migration with respect to the control and also with respect to the EFM. So the same thing is shown here in the curve, and this is a representative experiment from one patient, and this box plot instead shows how the trend is always the same, and the numbers are still significant when we look at 7 different patients. So furthermore, I decided to test, since my hypothesis is based on effect on protein content within the exosomes, I've added a further negative control, and it's shown here in this curve as in the dark blue line. And for this, I've been boiling the exosomes in order to denature the protein, and I've been treating the SECs25 as well as with a boiled exosome pellet, the same concentration, and we see that there is no effect. The curve actually overlaps with the control. So this validates my hypothesis that's based on some proteomics finding that some proteomic component within the exosomes is actually enhancing the cancer cell migration in this case. And in this box plot, I show the same experiment by adding also the AF exosomes and the AF EFM counterpart. So we can see how the treatment with adjacent fibroblast exosomes, by showing here in orange, is still significant versus the control. And there is no significant difference between AF exosome and CAF exosome treatment, although there is always a trend that I've been observing across my experiment. And here, this is the last experiment I've been conducting by also adding a treatment with CAF-derived microvesicles, which are the bigger vesicle fraction that's usually range from 1 micron. And we can see how there's no significant effect versus the control, and this effect is comparable to the one that's given by the EFM treatment. So moving forward on this, I've been evaluating also the effect of CAF exosomes on cancer cell growth, and similar results have been shown here. The exosomes are enhancing SECs25 growth, so stimulate proliferation, and this is significant versus the control, and also significant versus the EFM. EFM still has the treatment with the just media still has an effect versus the control, but it's way minor than the exosomal one. And I've also been observing that when SECs are treated with exosomal pellet, there is an activation of the MAP kinase pathway, which can explain also the effects that we have been observing here on cell growth and cell migration. But going a little bit deeper into the CAF exosomes, going back to the proteomic signature, I wanted to find exosomal enriched proteins only contained in the CAF. So for doing this, I started applying some filtering criteria to the full data set in order to select for those proteins that are enriched in the CAF specifically. So I took the list of exosomes and I enriched for CAF proteins that are exactly higher in the CAF versus the adjacent fibroblasts by giving a p-value of 0.05, then a fold change of 1.5, and then also the protein had to be higher in the CAF in at least 3 out of the 4 pairs that are being analyzing in the screening, in the proteomic screening. So lead me to a final list of 180 proteins, and we can see this protein here visualized in the volcano plot, where in the x-axis it's a ratio between CAF and AF, whereas in the y-axis is the p-value, and the dark color in here represents the protein has enriched in 3 out of 4 patients. So the 180 proteins that I'm talking about are localized in the area within the red lines and by the dark color. So then I applied the same filtering criteria also to the cell media as well as to the cell lysate fraction. And I came up with this 3 small lists of proteins that here we can see in the Venn diagram how they overlap within each other. And also there is 5 proteins that are commonly found enriched in the CAF versus the adjacent fibroblast across all the 3 fractions that have been analyzing by proteomics. And this is consistent, and we can see where those 5 proteins map here in the volcano plot. This work is still ongoing, and we're actually validating one of these proteins, so I won't show those proteins. We'll stay anonymous for today. I just want to show that I've done some further validation in the validation cohort of different patients in lysates and in a pool of exosomes for one of them. And we're actually validating this by immunohistochemistry in patients. So going back to the CAF exosome enriched protein signature, I took the small list of proteins that are enriched in the CAF, and here indicated in red, and I compared this to the one that instead are at the bottom of the list of this comparison, so they are enriched in the adjacent fibroblast. When I compared this to small lists of proteins and I ran a gene ontology analysis, this has been done by using the FIBROBLASTS software. It's one of the striking difference that I noticed immediately is the enrichment in the CAF exosomes for proteins that are involved in the metabolism and energetic pathways. And if I zoom more into this list of proteins, and those are mainly enzymes, and it's a list of 51 proteins, those are involved in different biological pathways, the metabolism of nucleotides, purine, lipoprotein, and glucose degradation, as we can see here in the graph. And this doesn't apply to the same comparison when the same comparison is done on the whole cell lysate. So it seems like there is something that's specifically enriched in the exosomes and in the CAF exosomes, and it's involved in metabolic pathways. And this heat map here shows in red the proteins that are precisely enriched in the CAF exosomes, and we can see the intensity of these proteins, and the AF fraction is much lower, and the same condition does not apply for the cell lysate, as I just mentioned by looking at the FIBROBLASTS analysis. So here it comes the next question, if do CAF exosomes stimulate tumor metabolic activity on SEC cells? And this also are experiments that are currently ongoing. I've been analyzing, evaluating glucose metabolism and mitochondrial stress test by using a CORS analysis, and this data is still preliminary, so I will not discuss it today. So just summarizing what I've shown you so far, mainly we've been looking at the effects of the proteome of the exosomes coming from fibroblasts, tumor fibroblasts, and normal fibroblasts, as well as the effect of exosome on oral cancer cells. But then to complete and dissect the signaling more in depth, I've also completed an analysis of the soluble proteins that are released by the fibroblast. And here, if we zoom in this fraction, we can see in this 3D plot how marked in red, a smaller list of 422 media-enriched proteins. And those are the proteins that are rich in the media versus the exosomes and the cell lysate. And here is the gene ontology analysis that shows the quality of this protein. Those are mainly extracellular proteins, secreted, etc. And so to complete this network, we also looked at receptors and plasma membrane protein on the recipient cells, so in this case on the oral cancer cells. And this has been done still by applying mass spec-based proteomics on 2 different cell lines, SCC25 and SCC4, and they're both derived from tongue cancer. The cell line has been grown till a monolayer, and then the monolayer has been coated with a colloidal solution of silica beads. Those beads are charged, and they electrostatically interact with the surface molecules present on the cells. And then the membrane fraction can be pulled down by pulling down the beads and by purifying the fraction by doing ultracentrifugation on an impedance gradient, actually. So in the end, what we get is a pellet of plasma membrane, which is enriched proteins, enriched surface molecules, as well as we also process by LCMS the homogeneous fraction, again, to have it as a background, more of like a control, to look at our enrichment. So this led me to identify 5,757 proteins. And this Western blot here is just a panel that validates how membrane proteins such as EGFR or CD81 are only present and enriched, actually fairly clean in the plasma membrane fraction versus the membrane depleted fraction, whereas intracellular protein are only present in the MD fraction and not in the plasma membrane. And if we do a gene ontology analysis of those proteins, again, we can see how the plasma membrane proteome of SEC cells, in this case, is enriched for cellular component in plasma membrane proteins for biological process. So, of course, there's like enrichment in cell adhesion, transmembrane transport, and when we look at molecular function, we see how we have transporter activity, signaling receptor, protein kinase, etc. So, and this is precisely what we want to look at, and we're interested in looking at. So the proteomic analysis of plasma membrane protein lead us to a smaller list of 1,500 plasma membrane enriched proteins, and this list of the plasma membrane proteome will be then integrated with the secretome that comes from the fibroblast. Just to remind you, is a list of 422 million enriched proteins, and so this will be done and is currently actually ongoing. By aligning those enriched lists to a database, that's how we started to proceed, of ligand receptor mediated signaling in human that has been recently published on Nature Communication. And so this is because we want to finally have a whole picture of the pro-tumorigenic signals of the tumor microenvironment, and to address IM4, I want to build a more comprehensive model that's based on a map of ligand receptor interaction of the tumor microenvironment in head and neck cancers. So just to conclude for today, I've shown you how I successfully isolate and characterize CAF and AF from tumors. I've been conducting in-depth proteomics analysis on 3 different fractions of the cells: the media, the exosomal fraction, and the whole cell lysate. I was able to highlight a CAF unique protein signature in exosomes, and also to validate one of the markers that can be in the future also utilized as a CAF specific marker of the CAF activated stroma, which at the moment we're currently lacking. And also how the CAF-secreted exosomes can stimulate SCCs25 motility and proliferation, and this is also a process that probably due to the activation of MAP kinase signaling pathways in the same cells. As future goals, I am analyzing and investigating the effects of CAF and AF exosomes on SCC's metabolic functions. And also, I'm building a MS-based ligand receptor repertoire of the tumor microenvironment in order to better dissect the signaling and have more insights into these tumors. So with this, that's all for today, and I would like to acknowledge my mentor, Dr. Thomas Kissinger, as well as the members of my lab that have been contributing directly and indirectly to this project. And our collaborators, Dr. L'Oreal, and people in our lab, Akira Angelna and Adrian Jan, for having realizing one of the cartoons, some of the nice cartoons that I'll be showing to you today, as well as my fundings, and of course, special thanks to Beckman Culture and Lab Roots for giving me the opportunity today to present this webinar. And thanks to all of you for your attention. I'll be glad to take any question.
Great. Thank you for that terrific presentation, Simona. Before we get started on the question and answer session, I would like to remind our audience that you can submit questions by typing them in the Q&A box at the lower left of the presentation window. Several good questions have already came in today. Our first question is, which markers are used to characterize the AF and CAFs?
Okay, so that's a very interesting question. I'll be mentioning something during my talk. The main markers that are used to characterize the CAF phenotype, so the CAF-activated, fibroblast-activated phenotype are a smooth muscle-actin protein, the alpha-SMA and the gamma, and also as well as some secreted proteins such as increase in collagen-1 production by CAF cells, and also it's known that CAF secrete proteinases such as MMPs, MMP1 and MMP2, TIMP1, and proteins that are involved in degrading the exosomal matrix and in creating the degradation of the basement membrane, as I showed you before. So those are the main markers known so far, but much to do. Much more needs to be done on the characterization of the cells.
Great. Thank you for that answer. Our next question that comes in is, did you do any proteomics on the resected tumors to assay the exosomes?
Yes, wide and in-depth proteomic analysis have been done on the exosomes, but not on the tumor counterpart. Well, the tumor has been analyzed as well because the whole cell lysate that we're looking at in the proteomics analysis actually comes from cells that have been isolated from resected primary tumors. So yes, the proteomic analysis is very broad and goes in depth and contains all the possible fraction, including whole cell lysate exosomes and secretome.
Great, thank you for that. Along the same lines, a question was, has lipidomics been used to characterize the phospholipid or lipid composition of these exosomes?
Oh, that's a very interesting question. Yes, lipidomics has been used to characterize exosomes, not particularly for this kind of tumors, but there is some lipidomics analysis still mass spec based on, for example, prostate cancer cells, PC3, and a few other cancer cell type. Of course, the lipidomic field is not as advanced as the proteomics, but yes, there are studies that actually have defined the lipidomics of the exosomes.
Terrific. I think we have time for one more question. Is there any influence of immune-associated molecules like chemokines on the tumor milieu on the exosomes of CAFs?
Oh, another very interesting question, and so there is the chemokines and interleukins have absolutely a key role in the development of this cancer, as well as in the paracrine activity that goes from the cancer cells to the fibroblasts and vice versa. And yes, there is a repertoire of molecules that are known to be secreted from the cancer cell, such as, for example, TGF-beta or interleukin-6. They're known to activate the fibroblast in the tumor stroma, as well as there are chemokines and interleukins that are also secreted from the fibroblast and goes the other way to the tumor.
Thank you again. Unfortunately, we are out of time today. I want to thank the audience for their interesting questions and participation in today's event. I would also like to thank our sponsor, Beckman Coulter, and of course, Dr. Simona Principe for making today's educational webcast possible, which will also be available for on-demand viewing through beckmancoulter.com and labroots.com. Thanks again. Goodbye.